31,000 reported. 938,000 people have recovered from this disease. So before going into the details of the topic, I would like to start with the very basic information. So what are the viruses? The viruses are microscopic parasites which are much smaller than that of bacteria. And they do not have the capacity to reproduce themselves outside of the host cells. So they are being considered as a non-living organism. So just to give you an idea of their size, how small they are, if you compare the measles virus with the Good evening, everyone. My name is Neetu Kalra, and I'm here to talk about the general characteristics of SARS-CoV-2 virus and measures to protect from the disease. So I hope you all are safe and staying at home. So let's begin. So if you look at the data in India, we have around 31,000 reported coronavirus cases and 1,000 deaths so far. In other terms, we can say it is about 0.76 deaths per million. And if you compare this data to that of United States, where the number of deaths per million is more than 175, definitely we are in better position. So if you look at the coronavirus cases worldwide, we already have the 3.13 million confirmed cases, out of which we already 938,000 people have recovered from this disease. So before going into the details of the topic, I would like to start with the very basic information. So what are the viruses? The viruses are microscopic parasites which are much smaller than that of bacteria. And they do not have the capacity to reproduce themselves outside of the host cells. So they are being considered as a non-living organism. So just to give you an idea of their size, how small they are, if you compare the measles virus with the bacteria, it is about 8 times smaller than E. coli bacteria. And if you look at the hepatitis virus, which is even smaller, it is about 40 times smaller than that of E. coli bacteria. So the figure here shown gives you the idea that DNA is of 10 nanometer, virus diameter shown here is around 100 nanometer scale, bacteria is 10 times more, and if you look at the human cell, it is 100 to 1000 times more if you compare it with the virus. So we are dealing with a very small entity. If you look at the structure, simple virion, which is a single particle of virus, it comprises of an inner nucleic acid core, which is surrounded by an outer casing of protein known as capsid. So the capsid basically protects the nucleic acid from any kind of enzyme which is produced by the host cells. So in some viruses, other than the capsid, they have the outer protective layer known as envelope. So the DNA or RNA found in the core of the virus, it could be either single stranded or double stranded. So this DNA or RNA constitutes the genome of the virus genetic information. So in general, the genomes of the viruses are very small because they essentially code for the proteins which is important for the necessary actions like replication, and most of the protein they utilize are of host cells. Coming back to the topic, in last year, November, December, an outbreak of novel coronavirus in Wuhan, China. Initially, it infected around 40 people. So those people actually developed the symptoms of pneumonia. So at that time, they had no clue that how many people were initially infected because the virus was novel and they had no means to diagnose the disease. However, they had some idea that most of the patients they had contact with the same seafood market in Wuhan, China. And eventually, more than 1400 cases were reported in Hubei province. So on February 11, 2020, the World Health Organization they announced an official name for the disease. The new name of this disease is coronavirus disease 2019, which is abbreviated as COVID-19. So CO stands for corona, VI stands for virus, and D stands for disease. So formerly it was referred as 2019 novel coronavirus. So why corona? Corona is a Latin word which means crown. So the spike glycoproteins which are recruited out from the outer surface of the virus, they form a appearance of a crown. That's why they gave this name as a corona. And this is not a new family of virus. It was first identified in mid 1960. So we know there are four main subgroups of coronaviruses known as alpha, beta, gamma, and delta. And these coronaviruses are family of virus that cause the respiratory illness. So if you look at the structure of the COVID-19 disease virus, which is known as SARS-CoV because of its similarity to the SARS-CoV virus, it has a spike like a protein which gives them the appearance of the crown. Then it has a membrane protein, nucleoprotein, envelope small membrane protein, and it consists of a genomic array. So now, as mentioned in the previous slide, the coronavirus have four subgroups: alpha, beta, gamma, and delta coronavirus. And the transmission occurs through both interspecies and intraspecies route. In case of transmission in humans, alpha coronavirus and beta coronaviruses are involved. And there is no data which talks about the gamma coronavirus or delta coronavirus. So the next most common question was from where did the virus come from? So the research group they came up with two possible scenarios for the origin of SARS-CoV-2 in humans. And this scenario was based on the origin stories for a few other coronaviruses which caused epidemics in human in past. The first one was Middle East respiratory syndrome known as MERS-CoV, and the second one was severe acute respiratory syndrome known as SARS-CoV. So this MERS-CoV was initially identified in Saudi Arabia in 2012 and it caused over 800 people's life in Middle East. In case of SARS-CoV, it was identified in 2003 in China and it killed nearly 650 people in China. So in both MERS and SARS, the humans they contracted the disease from an animal. It was civet cats in the case of SARS and camels in the case of Middle East respiratory syndrome. So similarly, in the case of SARS-CoV-2, the researchers suggest that animal was a bat. So initially, it was transmitted from the bat to another intermediate animal. possibly a pangolin, and from pangolin it was transmitted to humans. So there are total 7 coronavirus which infect the human. So out of those 7 coronaviruses, the 4 most common human coronaviruses named 229E, NL63, OC43 and HKU1. They did not jump from animals to humans. They rather chose human as their natural host. So the whole idea for these 4 coronaviruses was to spread their population. They were not interested in the pathogenicity. So that means the viruses, they opt for maximizing their spread rather than harming the humans. So this slightly explain why the coronavirus that are transmitted from animals seems to cause more severe disease in humans. But still the idea is speculative. So these 4 more common human coronaviruses, they cause mild symptoms, not like the rest of the 3, which rather cause the severe disease. So even if there are the phylogenetic similarities among SARS-CoV-2, severe acute respiratory syndrome, coronavirus and the Middle East respiratory syndrome coronavirus, indicating that they might have the common um, strain from which they are all originated. There are still some differences in the clinical characteristics such as greater transmissibility, relatively lower frequency of gastrointestinal symptoms, more patients with mild symptoms, and fewer fatalities in case of COVID-19, which distinguish it from rest of the two. So how contagious is the coronavirus? If you look at the average number of people infected by an individual, this graph shows that top, the measles virus here is the most infectious one, where you have the number in between 12 to 18. In case of novel coronavirus, the range is between 1.4 to 2.5. So if you have number more than 1, that means the disease is infectious, which is the case in case of COVID-19 as well. However, it is not that infectious like that of measles. So one infected person can roughly infect 2 to 3 more persons. That's what we think. Having this data in mind, the next questions which researchers asked was how this coronavirus actually enters inside the host cell and whether this SARS-CoV-2 virus is a man-made mistake or else it is uh, originated through natural selection. So in order to answer this question, one of the researcher, Kristen Anderson, an associate professor of immunology and microbiology at Scripps Research, and his colleagues and few other groups were involved in finding out the mechanism of the entry of this SARS-CoV-2 inside the host cell. So as I already mentioned that this virus contains the spike proteins, the glycoproteins that protrude from the surface of the virus. And basically this virus uses these spikes to grab the outer wall of the host cells and then enter those cells. So these researchers, what they did was they specifically looked for the structure and the sequence of the spike protein. And that also specifically to the grabber, which actually is a receptor binding domain, and the cleavage site that allows the virus to open and enter the host cells. So this kind of studied, they compared the sequence of the DNA of the receptor binding domain of this SARS-CoV-2 to other coronaviruses. And when they compared, they found there were few novel amino acids which were present in the receptor binding domain of the spike protein. And they found that this receptor binding domain, known as grabber, it can hook on the host cells. And basically, it binds to the receptor which is present on the surface of the host cell, known as ACE2 or angiotensin receptor 2. This receptor is involved in blood pressure regulation. And this attachment of binding domain of SARS-CoV to the ACE2 receptor is it's, it's very effective. So because of the results of their experiment and looking at this effective binding, they came up with this speculation that the spike protein, they are the results of natural selection and not genetic engineering. However, we still require more data to finally come up with any conclusion. So if you look at the life cycle of SARS-CoV-2 in host cells, as I mentioned, they use their spike protein and basically the receptor binding domain, and there is also a cleavage site present in the spike protein which helps them to enter inside the host cell. So this receptor binding protein goes and bind to the receptor ACE2, and once they are in the fixed position, uh, the fusion occurs and this whole virus goes inside of the cell because of endocytosis. And once it is inside, they will be uncoating, the RNA will be released inside the host cell, and now it will use the host cell machinery for the process of translation and replication. So remember I mentioned before that virus genomes are very small because they code only for the essential protein. So for rest of the processes, they depend upon the host cell machinery. So they kind of hijack the host machinery to produce the proteins of their interest and to replicate their genome. So after translation, which is the process of production of protein, they have now ready spike protein, envelope protein, membrane protein, and nucleocapsid protein. And simultaneously, because of the replication, they have now copies of their RNA genome. So once everything is ready, now there will be assembly and budding on rough endoplasmic reticulum. Finally, the packaging occurs inside the Golgi apparatus and in form of vesicles, and through exocytosis, the viral particles will be released. Now they are ready to infect the nearby cells. So once they are inside the cells, especially the host cells which express this angiotensin receptor, their only job inside the cell is to replicate themselves. Once they are in good number, the cells will release them. Cell may get burst and they will be released, and they are ready to now look for new cells. And finally, the whole organ will be full of virus. So how is the virus transmitted? The major routes of transmission of SARS-CoV-2 are believed to be person-to-person transmission through respiratory droplets. So once a person sneezes, it can produce up to 10,000 droplets. And if the infected person cough, it can produce 3,000 droplets. So these droplets, if they becomes airborne, they can stay in a air for a few hours. However, if it lands on a surface, depends upon the surface that for how long the virus can survive. There are some data which shows the presence of the virus for starting from 1 day to up to few days. So the person can get infected through this respiratory droplets or through close unprotected contact with any individual who has contracted the disease. So basically through both direct contact and indirect contact. So if you look at the common symptoms which appears because of this disease are fever, fatigue and dry cough. So according to the data, 80% of the persons they will have a mild disease, 13% will have severe disease, and rest of the 6% will have critical disease. So in case of the critical disease, the patients will develop the symptoms of pneumonia. They will have a problem in breathing and in some cases multiple organ failure, which may lead to death. So when do some symptoms of COVID-19 appear? So in general, it may appear in between 2 to 14 days. On average, within 5 to 6 days, you should see the symptoms. So because we know this SARS-CoV-2 is very much like of the rest of the two coronavirus, like SARS-CoV and MERS-CoV, which can survive on the dry surfaces for extended period of time, it is important to disinfect the surface and make sure to clean the surface properly. So in this case, the disinfectants which have been shown some activity against the enveloped viruses, those includes sodium hypochlorite, 70% ethanol, 0.5% hydrogen peroxide, quaternary ammonium compounds and phenolic compounds. So you have to make sure that you wipe the surfaces properly with one of these chemical disinfectant. So there is another very interesting terminology which you might be listening nowadays is about the flattening the curve. So what actually this flattening the curve means? So if you look at the curve, the y-axis says the number of cases and the x-axis tells you about the time since the first case. So in epidemiology, the curve refers to the projected number of new cases over a period of time. So if you look at this figure here, this graph, there is a steep rise in the number of cases, and that goes beyond the healthcare system capacity. So this dotted line is actually the healthcare system capacity which you might have in your city or in your country. Um, so if we don't take any protective measures, the number will rise exponentially and eventually they will go down because the people will start recovering from the disease or they might die because of the disease. So eventually it will go down. However, if you take a proper protective measures like staying inside the home um, and doing all those protective measures which is required um, for the prevention of the disease, then even if the number of people who are getting infected are the same, but it happens over a longer time. So this is within the healthcare system capacity. So this will give people better access to care. And also this will give a time for the researchers to do some experiment to come up with some ideas about the virus infectious activity, and other questions which are still not answered. The best way is prevention. And how can you prevent this disease? Is a few things you can do, which every individual can do, is wash your hands properly and more frequently, especially when you come um, from outside, and then you have to cover your mouth, your nose using a mask. Make sure you disinfect most of the instrument or equipment you're using at present in your inside your home. Avoid any kind of social gathering. Keep the distance which is required so that there won't be any, if there is any droplets which cannot travel. So you have to have the distance as it is mentioned. Do not touch your eyes, mouth, or nose, and cook thoroughly. So these are the few measures you can take in order to prevent this disease. Now, if you look at the treatment, we don't have any treatment present. Treatment in a sense, there is no cure for this disease, and basically the patients are treated for the symptoms which involve fever, cough, and shortness of breath. So, in case of mild cases, this might simply mean that you take proper rest or you take fever reducing medications. However, in those 6% critical cases, which includes pneumonia, where the patient have a lot problem in breathing, the treatment involves the ventilation with oxygen. So this ventilators basically they help to blow the air into the lungs through a mask or a tube which is directly inserted in the windpipe. So, how do we diagnose the disease? The diagnostic is based on the kit, which is developed by the Center for Disease Control and Prevention, and it's known as 2019 Novel Coronavirus Real-Time Reverse Transcriptase PCR Diagnostic Panel. So this test is basically intended for the use with upper and lower respiratory specimens. So these specimens can be taken, and once you have this specimen, both for epidemiological surveillance and for the diagnosis, this kit is being utilized. Once the symptoms are known, which is called overt disease, then proper staging, prognostication, and therapeutic monitoring is also required. In this case, few other biochemical assays are done in order to confirm the presence of the disease along with real-time PCR diagnosis. And once the recovery occurs, you still need to do the testing in order to make sure that the patient is now virus free before you ask the patient to go back. So this real-time PCR assay is a probe-based assay where you use the probe which is specific for your DNA or RNA sequence. This probe contains a reporter dye as well as the quencher dye. Other than probe, this assay also have the primers which are very specific sequence and goes and bind to the template DNA or RNA. So once the primer finds its complementary sequence, there will be a polymerase enzyme which will extend the primer sequence. And once it reaches near to the probe will be hydrolyzed. The reporter dye will be removed. Now it is not quenched by the quencher dye. So now there will be a fluorescence which will be detected, and that will tell you the presence of that particular sequence in the particular sample. So in the case of coronavirus, SARS-CoV-2 real-time RT-PCR nucleic acid detection kit, it is based on the PCR method, which uses a fluorescent probe, like I mentioned in the previous slide, and the primers, which detects three specific regions within the novel coronaviruses. So this novel coronavirus's specific region lies in the nucleocapsid protein N gene. So this N gene sequence is basically distinguished. These are the regions which will distinguish it from the previous SARS-CoV virus, right? Because we don't want to um, get confused with the other virus. We want to make sure that whatever we are detecting here is only SARS-CoV-2. So this kit includes three primer probe set corresponding to those used in the CDC real-time diagnostic panel. Okay? So they already have given the guidelines that which region specific primers you have to use, because those regions are the novel regions which are present in the SARS-CoV-2. So once you have the primer probe sets, this called N1 and N2, that detects specifically the SARS-CoV-2 sequence. So there won't be any false positive result, right? And because this SARS-CoV-2 is RNA virus, you need to make sure that first you extract the RNA. After extracting the RNA, you convert the RNA to cDNA using reverse transcriptase enzyme. Once you have the cDNA, now you have the primers which are specific to the N1 and N2 region of that particular gene. And once you have that primer, the primer will go and bind to the region. If the sample contains the virus, primer will go and bind. Probe is there, and this probe set will finally, you will have the fluorescence which will be detected. So in addition to the N1, N2, we also have a set of primer probe for the housekeeping gene in order to make sure that there is no problem with the reaction. So it acts as an internal reference to monitor the sample correction, that the RNA is extracted properly, the quantity of RNA is fine, and the amplification is taking place. So once you have the positive result, this gives you that the patient is already infected. So this should be done for all the symptomatic which already have the symptoms for the for the disease, as well as for epidemiological surveillance in a community. Another important therapy, which is known as convalescent plasma therapy. In this case, what we do is we take out the plasma. So basically we remove all the blood cells and we only take the plasma which contains the protein, antibodies, minerals. Now because you have the antibodies, especially from patients who have recovered from this disease, we are only talking about the patients who have recovered from the disease, and we take out the blood, we extract the blood plasma. And now we want to use this plasma to treat other patients who have contracted the disease. So this experimental therapy, this goes back 100 years, having found use to different other diseases like Spanish flu, as well as the 2009 H1N1 and SARS and MERS also. So the whole idea is that we are using the immune cells of the patient who already have recovered, no more suffering from the disease, because he has developed the antibodies to fight with these viruses. That might also help the other patient to fight back. However, this is still not sure whether it can be used for the treatment, as 2-3 days back only the government of India has announced that it cannot be used as a method to treat the patient. So outside of India, there are a couple of places where they're doing the clinical trial using this plasma therapy. So let's hope for the best. Another important thing, like other than this real-time PCR kits, CDC is also supporting the global efforts to study and to learn about the virus that causes COVID-19 in the laboratory. And how? Because they are growing this virus in cell culture. So once you have the good number of viruses outside in cell culture, now you can use different antiviral drugs, right? And you can find out that how effective these medications are against of the SARS-CoV. Basically, you're focusing in the field of antiviral research. Another important thing is the pathogenesis, like how actually this SARS-CoV-2, we know we have some idea that it originated in China, and the initial animal was bat. From there, maybe pangolin, and from pangolin to humans. We don't have the clear answer. We don't know what are the host organs which are infected more, how many viruses are present inside the host, and the severity of the illness which is causes in the host. So basically, in order to answer these questions, it will be helpful if we have the viral culture with us. Another important thing, like I mentioned before, also that once it reaches to the different surface, these viral particles, they can survive there for different time period. So we can also study about the virus stability, right? So this is also an area of research where you try to find out the stability of the virus. Recently, we heard that because of the heat and high humidity may reduce the number of virus. Let's see what happens. But other than the data which is coming from US or China, now the Indian government, all the funding agency, they are also kind of ready to fund the projects to study the various aspects of the novel coronavirus. So we are all together into it and looking for uh, various ways to come out of this. Now, the most important thing is when will we come up with a vaccine specific for novel coronavirus, right? So, if you want to look at the current perspective, we have already seven front runners, all of which have entered human trials. So, that's a good news for us. However, the process of development of the vaccine, it's not a easy job. As you know, the Rome was not built in a single day. So same goes for the vaccine. Sometimes it takes like, usually it takes a decade because you have to go through different human trials, phase trials, and then you have to look for the safety, the side effects, and of course the ability to induce immunity. But in case of novel coronavirus vaccine, we are looking for the for next couple of years we should be able to have the vaccine in our hand. So one of the British vaccine, it's already in the human trial. They use a harmless virus. So you basically use the attenuated strain of the virus and then you introduce the genes that produce the novel coronavirus spike protein. Like I mentioned, those are the protein, those are the region which makes it different, which distinguish them from the other coronaviruses. And once you inject that virus containing that spike protein inside the human body, that forces the immune system. Like this is not something new for us. It's not the part of the human body. So they will build cells that disable the spike proteins. So that ability that would protect against a future novel coronavirus infection. So then you will develop an immunity, you will develop the antibodies, which in later exposure of the virus will be ready to fight it back. So we are hoping to have the vaccine very soon, not only outside of India, uh, but also in India. We are there are a few companies which already have started clinical trials. So let's see. Now I would like to finish this talk with this quote from Marie Curie that nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. It's true for us, and there are a couple of links which you can click and have more information related to coronavirus. And stay at home and stay safe. Thank you.